Hello everyone and welcome to this audio podcast in this series, the ZtormCrux Continuum. This is the Shientist, and this is an audio podcast, a part of the Storm Robotics production, and this is part 3 surrounding the ZtormCrux Continuum, and the sort of final part in this particular series. And what I thought I'd do is provide an overview relating and regarding works that had been developed in around 2007 surrounding transplantation immunology versus neuroendocrinology and psychology. And in this works or in this original works, the outline around this in its original in the original context is in 3 parts. And so what I thought I'd do is provide an overview surrounding each of the parts 1, 2, and 3 surrounding these works, in and around these works, and in context of providing a hypothesis, a background, some aims or objectives, some context around the citation, some context around proposed research questions, and then in parts 2 of the original works, there's context around sort of authors who had developed particular sort of models in context of clinical medicine, as well as a link to sort of aspects surrounding laboratory medicine, and then some context around a literature review, some research methodology, and some context around sort of future research, some aspects in surrounding sort of alongside with the sort of research methodology, some context around sort of some some aspects surrounding research and investigation in context of prospective investigations, and then some context surrounding some discussion, a discussion and conclusion, and wherein the original works are put in context of both a sort of discussion and conclusion, alongside some sort of final points. So to begin with, I am going to provide an overview. I'm going to provide sort of a hypothesis surrounding these works in and around transplantation, immunology versus neuroendocrinology and psychology. So to begin with, the hypotheses. So the immune system still remains to be a complex entity when considering patients for renal transplantation. Transplantation remains the most feasible route of treatment for a form of renal replacement therapy for those who are identified as in CKD or end-stage renal disease, or in and against other forms of renal replacement therapy, for example, peritoneal dialysis or hemodialysis. But because of the lack of donors, the MHC major histocompatibility complex and aligned to human leukocyte antigens or HLA inconsistencies, for example, renal failure or chronic kidney disease or end-stage renal disease, still remains a global challenge in this context. The immune system and chemokine receptors may not be the only sort of markers involved in rejection episodes. In some works by Mohammed in 2006, where there was in works where methods for investigation had informed that if there is positively expressed migration in context of particular immunological components, for example, in aligned with stress cellular adhesion molecules in an isolated T cell culture system, it could imply that psychological stress is another complication post-transplantation. Now, as part of this hypothesis, this work seek to emphasize literature or identify literature and emphasize on the transplantation and psychological theory, and also endeavor to perceive whether there is a particular system which dominates in patients who may require transplantation. So, for example, the immune system, neuroendocrinology, or psychology in context of mental attitude, by looking more in depth, and intricately in and around the different pathways. In and around the pathways and the different physiological systems and of course in and around the different systems. So it is hypothesized that one of these systems is more dominant pre-transplant than the other, predisposing patients to acute allograft rejection post-transplant. It is also proposed that that investigations involving psychological stress and the neuroendocrine system should be included with sort of more current immunological concepts associated with acute allograft rejection to establish more prominent and potential links. So in line with the hypothesis, the next sort of aspects that are to be highlighted are in context of the background surrounding this works around transplantation immunology versus neuroendocrinology and psychology. So the immune system protects from a physiological standpoint from infection or infections by pathogenic organisms or through pathogenic organisms as well. It is composed of a complex constellation of cells, organs, and tissues arranged in an elaborate and dynamic communications network. The immune system is, in its simplest form, a cascade of detection and adaptation culminating in a system that is remarkably effective. Leukocyte and lymphocyte migration in context from an immunological context and standpoint, and also referred to as trafficking from peripheral blood into affected tissues, is an essential component of the inflammatory reaction to virtually all forms of injury, and it is an important factor in the development of many diseases. So chemokines as markers help to control the selective migration and activation of inflammatory cells into the injured tissue or allograft. Chemokines and corresponding receptors or receptor ligands migrate during and when there is inflammation. And so in and around this, I thought in and around this in the original works, there's an overview in regards to the components of the immune system. So as part of the immune system, there's the innate side, and there's the adaptive side. Now the innate side regards a response where it is non-specific. Exposure leads to immediate maximal response. Cell-mediated and humoral components are involved. There's no immunological memory as part of the innate system. And then it's found the innate system is the innate component of the immune system is found in nearly all forms of life. Now, the adaptive side and the adaptive component is where there's pathogen and antigen specific response, a lag time between exposure and maximal response, cell mediated and humoral components, as in the case of the innate side and innate component of the immune system. And then exposure leads to where there's immunological memory established, and also found only in endured vertebrates. As part of this, there's also as part of the components of the immune system. In this works, there's an outline to be made in regards to hematopoietic stem cells. So the immune system starts with hematopoietic, from Greek blood-making stem cells. So hematopoietic is terminology that stems from Greek terminology known as blood-making, and these stem cells differentiate into the major players in the immune system. So, for example, granulocytes, monocytes, and lymphocytes. The stem cells also differentiate into cells in the blood that are not involved in the immune in and through immune functions, such as, for example, erythrocytes, where in context of red blood cells, and megakaryocytes for blood clotting. And then also stem cells continue to be produced and differentiate throughout individuals a person's sort of lifetime. In the original works, there is a schematic or an illustration that summarizes the process in context of hematopoietic differentiation. Also in these original works, and as part of the sort of background around this, there's a really informative paper by Rossi and Zlotnick from 2000, which highlight works surrounding chemokines and receptors. Now, in and around as part of this works, I thought in and around this works, there's in the original works here, there's a schematic that sort of provides an overview on surrounding the biology of chemokines and receptors. So in this context, I'm going to look to provide a sort of brief overview, surrounding the schematic. So chemokines, as aforementioned, are involved in generally in lots of different processes surrounding the immune system and part of the immune responses. Now, chemokines are involved in the development of T helper cells type 1, T helper cells type 2. They're involved in the um, in the uh, uh, in the development around in and around angiogenesis and angiostasis. They're involved in metastases, involved in cell recruit, in particular cell recruitment. They're involved in inflammation. They're involved in lymphoid organ development, and involved in lymphoid trafficking, involved in wound healing. Now, in Rossi and Zlotnick's work in 2000, they highlight the they highlight more of an in-depth perspective surrounding biological functions of chemokines and chemokine receptors. Um, and so, as part of this, also, um, Uh, this then leads into an overview surrounding the link between the central nervous system, the immune system, and the endocrine system. And so, in this original works, there is an overview that links. There's another schematic that highlights a link between the nervous system, the immune system, and the endocrine system. And where there's a link generally, there's a sort of an integrative view of health and disease. And this schematic outlines the general relationship between the central nervous system, the neuroendocrine system, the immune systems, with health and disease. And the model, the schematic, sort of incorporates the concept that is referred or has been retrospectively referred to as the psychoneuroimmunology concept, or P&I concept. So in and around this, and continuing in context of the background, so many researchers have investigated and believe that physical health may be influenced by psychology through a variety of direct and indirect means. There is some evidence that for example, stresses, or those sort of influences that are linked between stress, depression, and anxiety, for example, can directly affect physical immunity through production of stress hormones such as catecholamines and glucocorticoids. Although research or the literature sort of goes where well-renowned authors, for example, have sort of debated between different sort of different components between the immune system and the endocrine system or neuroendocrine system. There is also some indication that certain psychological states may lead to faster disease progression in particular diseases. So, for example, the human immunodeficiency virus, or HIV, and cardiovascular disease or cardiovascular health through these direct biological mechanisms. So, emotional states may also indirectly affect disease processes through influence on sort of different health behaviors. So health psychology incorporates another particular focus or model called the biopsychosocial model. Now, so this model of medicine is a way of looking at the a mind and body of patients or individuals as two important systems that are interlinked or intertwined. So the model was developed by initially or originally by a psychiatrist known as George Engel at the University of Rochester, and George Engel first published works around this the biopsychosocial model in 1977 in an article of Science. The biopsychosocial model draws a distinction between the actual pathological processes that cause disease and the patient's perceptions of health and effects on it in context that is referred to as illness. And looking at entities based on the immune system, neuroendocrinology, and psychology tend to form the basis will form the basis of this work throughout. So I'm now going to take some time to highlight some aspects surrounding the aims around this works. So initially to primarily put forward literature surrounding the immune system, neuroendocrinology, and psychology that relate to transplantation, to identify whether there is a dominant system which causes rejection episodes, or in particular acute allograft rejection episodes, to see that transplant rejection is understood from various perspectives, and not solely on the immune system, and to see whether a collaborative approach in immunology, in transplant immunology, neuroendocrinology, and psychology is warranted to achieve T-cell tolerance post-transplant, and to see whether a holistic approach is required in patients awaiting transplantation, and also an effort to put forward potential future research projects or research questions which may be the route to a better understanding between the different aspects and focuses. So in order to do this, or to put forward some of the actions highlighted in the aims, the next part of this works is to highlight would be to highlight the citation aspects. So the citation aspects around this work involved a few particular aspects, wherein there was a list of main sort of terminologies that had been regarded as a link between different specialties or specialisms, and then as part of the sort of citation aspect, there was a sort of a process or a methodology, if you like, to actually implement the overview in relating and in regards to the different literature review sort of outputs. So for example, some of the terminologies, or the terminologies that had been implemented, or at least listed initially, included the immune system, renal transplantation, psychological stress, health psychology, clinical health psychology, psychoneuroimmunology, George Engel's Biopsychosocial Model 1977, Selye General Adaptation Syndrome 1936, cytokines, neuroendocrine, the chemokine receptor, cellular adhesion molecules, mental attitude, leukocyte and lymphocyte distribution, and quality of life. Now, as part of the list, the citation search was not the citation or the citation search, sorry, was not necessarily in the order of the words that have been read out. A combination of these words or terminologies were listed, and a majority of literature identifies was had been from using the PubMed search engine, and citation from 1987 to 2007 was implemented, and the main literature review or at least in the subsequent parts of this audio podcast, the main literature review outputs were from 1990 to 2007. So in part of this, as part of this this works or these works, the next part of this works is to highlight some proposed research questions in aligned to the literature, and again according to transplantation immunology versus neuroendocrinology and psychology. So, for example, one of the main questions to begin with was, how does psychological stress associate with transplantation and neuroendocrinology? Another question was research question was immunology versus neuroendocrinology. What does this mean? And another question, research question, was, how does this relate to transplantation? And another research question was, can T cell tolerance post-transplantation be achieved by better understanding on the surrounding the immune system, neuroendocrinology, and psychology in context of a link between the different systems? And could another question is, could a combined approach be could a combined approach be important to attain T cell tolerance? And so now, the original works here have also included is a sort of an overview of Selye's General Adaptation Syndrome or General Adaptation Model. In part of this, I'm going to provide a sort of overview of the General Adaptation Syndrome or General Adaptation Model. Selye was a physician and an endocrinologist, and it was around 1936 where Selye developed the General Adaptation Model or General Adaptation Syndrome. And what this was, so in this original works, there's a schematic that links different aspects and that surround the General Adaptation Model or General Adaptation Syndrome. So to begin with, there can be, for example, a stressor, and then from where there's a stressor, the hypothalamus gland and pituitary glands are activated, And then once the hypothalamus gland and pituitary gland are activated, the adrenal glands are then activated, and then there's a response. There's a link and a response between the gut or the stomach initially, the thymus or lymph nodes, the thymus and lymph nodes, and then the different types of white blood cells in context of, for example, where individuals are in either a stagnated, or either stagnated, or they are in a form where they are initiated to respond to a particular action, and as many would know, this relates to the in context, at least in some contexts, relates to is in regard to the fight and flight response. So Selye developed the General Adaptation Syndrome, or General Adaptation Model, in and around the time he was around. He was a physician and endocrinologist from 1907 to 1982. In line with this, as well, in line with this, it also in this original works, I thought alongside Selye, General Adaptation Syndrome, or General Adaptation Model, I then in this works have there's an overview to be put, an overview in regards to George Engel, who was a famous, who was a famous sort of famous for developing the biopsychosocial model in 1977. Dr. Engel's work can be viewed in accordance to Dr. Selye General Adaptation Syndrome from 1936, and it is perceived, or there's a perspective that both Dr. Engel and Dr. Selye achieved an immense foundation in aspects of medicine. Dr. Engel, for how to approach patients in context of the biopsychosocial model and a general theory of illness and healing, and also before Dr. Engel's passing, he was beginning investigations that would document the correlation of loss with the onset of variety of diseases. And again, Dr. Engel had a particular focus in around psychiatry. I'm now going to provide a sort of an overview that links the literature, or that links the literature with the sort of the premise of these original works. So to begin with, I'm going to highlight the following. The following is to be is what I'm going to highlight. So a large number of clinical and experimental observations indicate that immune responses may be modulated by the central nervous system, or CNS. It was Neville and Limol in the 1990s or 1990 where they informed where works informed that the immune system and the central nervous system are known to communicate through endocrine and the autonomic nervous systems. Nouveau and Lamothe informed that a complete regulatory loop between immune and endocrine systems exists. It was Wayne and team in 1990 who informed that there is a molecular basis for bidirectional communication between the immune and neuroendocrine systems. In this context, there are four sort of sub aspects. So number one, cells of the immune system can synthesize biologically active neuroendocrine peptide hormones. Number two, immune cells also possess receptors for many of these peptides. And number three, neuroendocrine hormones can influence immune function. And number four, lymphokines or lymphokines can influence neuroendocrine tissues. In and around this, the recognition of such stimuli is converted into information in the form of lymphokines or lymphokines, monokines, hormones, and message received by neuroendocrine tissues. On the other hand, nervous system recognition of stimuli can also be converted into chemical signals, signals that can be relayed to immune cells, resulting in physiological changes. It was Weyant and team in 1990 that also highlighted that the pathophysiology associated with infectious agents may be related or regarded to the types and amounts of hormones produced by the immune system. In part by the immune system, but also as part of the endocrine or neuroendocrine neuroendocrine systems. So prospective studies should be put forward to look at these in relation to transplant rejection episodes, specifically acute allograft rejection episodes. And it was Bezdovsky and Delray in 1991 who also informed that such interactions imply the existence of afferent messengers derived from the immune system, such as lymphokines and monokines, capable of integrating immune neuroendocrine circuits. And aligned to this as well, at the time of the peak surrounding the immune response to, for example, innocuous antigens, as well as following inoculation of infective agents, endotoxins or tumor cells, immunological cells, release cytokines which can stimulate the pituitary-adrenal axis. The acute effect of these cytokines is mediated by stimulation of the release surrounding corticotropin releasing factor from the hypothalamus. It was Grotowitz and team in 1991 informed that conditioned immunosuppression using a taste aversion paradigm have been demonstrated in a number of laboratory models. The team informed and reported that survival of allografts, heart allografts, particularly in rat models, rat laboratory models, can be prolonged by behaviorally trained immunosuppression using, for example, cyclosporin A or CSA as a stimulus in taste aversion conditioning. And conditioned animals received saccharin as the conditioned stimulus paired with an injection of cyclosporin A or CSA at 10 and 6 days before transplantation. Now, in line to this, these experiments suggest that These experiments inform or suggest that behaviorally conditioned immunosuppression may be important clinical implications as an adjunct to drug treatments in transplantation medicine. It was Growowitz who and team in 1991 who informed of that particular sort of outline. It was in 1992 that Grotzel and Sridharan informed that further knowledge based on the advantages in multi-system integration of multi-system integration of function in host defense may reveal other novel mechanisms of cellular communication and biological adaptation. It was Ottaway and husband in 1992 who informed that many of the alterations in immunity resulting from the central nervous system activity may be explained in terms of changes in lymphocyte migration patterns in response to endocrine signals, neural signals through transmitter release or direct contacts between nerves and cells of the immune system or surrounding the immune system. Disciplines ranging from anatomy to psychology revealed the immunosuppression as the target. The immune system revealed the immune system as a target of brain and endocrine signals. It was in 1993 that Kropotnick's findings inform that the immune system is active even in a bidirectional feedback loop. And today, the immune system is no longer regarded as an autonomous and scientists begin to see the emergence of other paradigms that are linked. So, for example, as some of those that have been aforementioned, but also the psychosomatic paradigm as well. It was Klein in 1993 that informed the cellular molecular basis from the observed modulation of host resistance is not fully understood, but involves altered functioning of both T lymphocytes and cells of the hypothalamic-Pituitary-Adrenal Axis. It was Klein also in 1993 wherein there was works involved where works involved the sort of altered production of cytokines and hormones produced by the immune system and the brain. It was works by Wainant and Blalock in 1995 who hypothesized that the relay of information to the endocrine system represents a sensory function for the immune system, wherein leukocytes recognize stimuli that are not recognizable by the central and peripheral nervous systems. So, for example, bacteria, tumors, viruses, and antigens. It was also Wainant and Blalock in 1995 who informed that future investigations into the physiological role that cytokines and neuroendocrine hormones have in these systems will be of considerable intrigue for both immunologists and endocrinologists. It was Mohammed in 2006 who informed briefly in regard to Shedlowski and Schmidt's work in 1996 that at the same time the stress hormones can change immune cell behavior and the activity of the immune system, but the general dilemma is that not enough is known in regards to precisely which hormones are more or less secreted, and it would seem dependent upon circumstance. Aligned to this, anger and frustration are classed as acute stress and will generally cause an activated immune response, but not the same as, for example, exhaustive exercise or physical activity. And so acute physiological, sorry, and so acute psychological stresses and physical activity have been shown to transiently enhance immune responses. Physical activity also causes an increase in beta endorphin, and because of beta endorphin, it may be this endocrine that suppresses T cells to an extent. After all, positive stress is generally good for the for it's generally good for physiology, from a physiological standpoint. And so this is also why an understanding of mental attitude is generally central. Other neuroendocrines need to be better established in regard to transplant acute rejection, or AR. AR episodes with primary immune components. It was Perner and team in '97 who highlighted, in terms of cardiovascular, endocrine, and immune responses, acute high intensity aerobic physical activity stress may be considered as a subcategory of stressful active coping. At the same time, neuroendocrine responses include increases in catecholamines as well as elevations in cortisol under high but relatively low sympathetic activation. Immune system responses include increases in natural killer cell number and cytotoxicity and suppressor or cytotoxic lymphocytes. Chronic life stressors, for example, influence acute cardiovascular, endocrine, and immune responses to acute stressors as well. So the comparison between a stressor such as transplantation, and where there's psychological stress pre-transplant and aspects leading to transplantation, for example, from a surgical standpoint to physical activity, may be important, may be an important similarity. So, for example, as sort of highlighted, acute psychological stresses and physical activity have been shown to transiently enhance immune responses. At the same time, physical activity also causes again an increase in beta endorphin, and because of the beta endorphin, the important immune units or components stay blunted. But in pre-transplantation, during stressful durations or stressful periods, there is no beta endorphin, and so the immune components such as chemokine receptors, CD8 T cells, natural killer cells may influence acute allograft rejection post-transplant. It was Ferenczak and Sterilanova in 1997 informed that neuroendocrine system influences that modulate neuroendocrine influences that modulate the immune function mainly include mental and physical stress. It was Danza in 1997 informed there is now evidence that there is evidence that in the same way that different perceptions of stress have different physiological consequences, different ways of coping and stress also result in different consequences on immunity, and the nature and outcome of which depend on the type of immune response. It was Webster in 1997 who demonstrated or informed that glucocorticoids and catecholamines favor T T helper type 2 cells over T helper type 1 cell or immune cells and mediators by controlling the production of specific key regulatory cytokines. It was Gaillard in in 1998 that informed the immune and neuroendocrine interactions are involved in numerous physiological and pathophysiological conditions and seem to play an important role to maintain homeostasis. It was in this works also the mutual information circuit inside of the immune, nervous, and endocrine systems suggest that they all form the superior system with pivotal regulatory role in living organisms. It was Ferencek in 1998 who informed that overall the recognition of the immune system as a sixth sense may ultimately provide a new understanding of physiology required for disease and stress involving immune, endocrine, and neuroendocrine communication. It was Klapek in 2000 who informed that through major question, though major questions remain, it appears reasonable to include the impact of anxiety or depression and possibly other psychological states among factors that may affect the net state of immunosuppression in transplanted recipients. It was Hennig in 2000 wherein through investigations surrounding mechanisms of changes in lymphocyte numbers after psychological stress. And within research sort of strategy, Hennig and team in 2000 found or identified that it became evident for catecholamine to increase upon stress and found it is of a major importance for the frequently observed mild leukocytosis and where there's an increase in the types of leukocytes. In another investigation, Hennig and team in 2001 demonstrated that cortisol seems to play an important role in stress-induced redistribution sort of patterns of CD8 cytotoxic T cells. This, however, can be explained by different migration processes between those cells, for example, different targets of migration and therefore different glucocorticoid influences on target tissues. It was Gaillard in 2001 who informed the crosstalk between the immune and endocrine systems is important to homeostasis, since the interactions can produce various appropriate adaptive responses when homeostasis is threatened. Recent investigations, or at least when this research was initially composed, recent investigations indicated that physiological levels of glucocorticoids are immunomodulatory rather than, for example, solely immunosuppressive, causing a shift in patterns of cytokine production from a T helper type 1 to a T helper type 2 pattern. So in line with this, this bidirectional communication is possible because both systems share a common sort of chemical language that results from a sharing of common ligands. So, for example, hormones and cytokines and specific receptors. Cytokines are important partners in this crosstalk. Cytokines tend to play a role in modulating, at least play a role in modulating aspects surrounding the hypothalamic-pituitary-adrenal axis responses at all three levels. So, for example, through the hypothalamus, the pituitary gland, and the adrenals. Acute effects of cytokines are produced at the central nervous system level, particularly the hypothalamus, whereas the pituitary and adrenal actions are slower and are probably involved during, for example, prolonged exposure to cytokines, such as during chronic inflammation or infection. It was Hubbard's in 2003, wherein the field of psychoneuroimmunology, or PNI, as a field of research, who highlighted the complex interactions between the central nervous system, endocrine, and immune system, and how behavior or stress can modify can modify interactions. Hormones, peptides, and neurotransmitters regulate the immune system, and understanding the complex interaction of these systems still remains a challenge for the 21st century. George Engel's biopsychosocial paradigm is perhaps one of the main models wherein dealing with most human health disorders. It also encourages broader reading into areas not traditionally associated with particular therapies. Pathogens, tumors, and allergens are detected with great sensitivity and specificity by the immune system. It was Blalock in 2007 informed that as a sixth sense, the immune system is a means to signal and mobilize the body to respond to these types of challenges. And also, one must acknowledge that research in and around transplantation should not be restricted to single organs, and that the psychoneuroimmunology concept may have a role in future investigations. This may produce a better understanding of the immune system, and it may be a means of attaining T-cell tolerance post-transplant as well. So now I'm going to take some time to highlight some context surrounding proposed sort of research methodologies in and around these works. So most of the investigations that are to be highlighted will surround sort of a premise around chemotaxis assays, so aspects surrounding laboratory medicine as well. So chemotaxis is a method for studying or investigating transport or migration towards a concentration gradient of chemoattractant for leukocytes. So, for example, neutrophils, monocytes, and lymphocytes, or other migratory cells. An upper chamber containing a suspension of cells is separated by a membrane from a lower chamber containing medium with chemoattractant. Chemotaxis of the cells from the upper chamber into the lower chamber occurs over a predetermined duration. The cells in the bottom part of the chamber may be counted directly when using a homogeneous population of cells, or may be stained for defined markers when assaying a mixed population of cells and analyzed through and by flow cytometry. And so, in line to this, using a T-cell culture system in isolation may be helpful to observe chemotaxis for example, number one, chemokine receptor cellular adhesion molecules and monoclonal antibodies, and use of flow cytometry analysis to identify migration. Measuring stress neuroendocrine levels pre and post transplant would prove admirable investigations. And number three, the idea of monitoring stress levels in pre-transplant duration or period would be of obvious significance and importance. Number 4, a study with chemotaxis assays could potentially observe catecholamines with repeated psychological stress, and this will potentially allow to identify how neuroendocrine hormones affect chemokine receptor migration. Number 5, observing psychological stress in pre-transplant stage with for example health professional interaction exploration. So observing placebo variables with this may be advantageous. Number 6, a study that incorporates chemotaxis and monoclonal antibodies with neuroendocrines post-transplant would be intriguing. Number 7, comparing some of the above sort of examples in pre-transplant phases would again observe chemokine receptors and neuroendocrines without monoclonal antibody therapy. Number 8, to examine the effects of acute psychological stress and exhaustive physical activity on chemokine receptor migration. And number 9, studies or investigations with natural killer cell activity or NKCA and cortisol with chemokine receptors in pre and post-transplant phases may also be noteworthy. And number 10, conducting investigations that relate or regard acute chronic stress, cytokine activity, and anxiety or depression may show significant correlations between the immune system and mental attitude. Number 11, measuring neuroendocrine function with the hypothalamic-Pituitary-Adrenal Axis and system and chemokine receptor migration in pre and post-transplanted recipients may well be valuable. And so, as some of the investigations have some of the sort of methodologies aligned to particular sort of questions, if you like, have been highlighted. The next sort of overview is here would be to provide a sort of an overview or perspective surrounding future research, further research and investigations. So, or at least, and also in context of the literature. So it was Al-Arabi and team in 1997 informed that pre-transplantation plasmapheresis together with immunosuppressive agents, so for example cyclophosphamide and corticosteroids is useful and can be helpful in the removal of human leukocyte antigen antibodies in immunized patients awaiting renal transplantation, for example, and it can be considered a valuable approach to increase the chances or the probability of a successful transplantation. In one investigation by Nafar and team in 2006, it was suggested that plasmapheresis with or without other therapeutic measures may have a role in the salvage of allografts with early acute dysfunction that are resistant to conventional therapy. And the team in Nafar team in 2006 concluded that allograft survival in patients with acute humoral response who respond to plasmapheresis can be comparable to that in other renal recipients. Having investigated and delved into the literature, in vitro investigations using interleukin-15, interleukin-2, interleukin-15 receptor alpha, and stress cellular adhesion molecules and relevant monoclonal antibodies, for example, anti-CD25, should be investigated with chemokine receptors such as CCR5, CCR7, and CXCR3 in a T cell culture system to see to identify if this affects T cell migration post-transplantation. Using peripheral blood mononuclear cells or PBMCs may well be advantageous. Again, using chemotaxis assays with flow cytometry would aid to give graphical data, and so linking specific endocrines such as cortisol, ACTH, and glucocorticoid hormone with the above would prove advantageous respectively. Stress may have specific effects with regards to T cell migration post-transplant, and instigating migration investigation is one of the most helpful ways to look at sort of the patterns. And so, in line with this, there are some aligned with this, there are some particular additional research questions that are important, and then in linking with the sort of discussion and conclusion around these original works. So one of the questions is, does the immune system serve as a sensory organ? And again, has been highlighted in literature retrospectively as a sixth sense. Another question is, why is it a challenge to attain T cell tolerance post-transplant? Another question is, should there be more efforts in the understanding of all systems and where there's a link between the immune system, neuroendocrine, and the sort of psychological aspects? And another question is, how do investigations in physical activity relate to transplantation sort of more specifically? And so this then brings this works into the sort of discussion aspects. So renal transplantation is generally the preferred form of renal replacement therapy for patients or individuals with in CKD, chronic kidney disease, or end-stage renal disease. In the pre-transplant period or duration, there are various factors that have to be taken into account. The notion of mental attitude or psychology and neuroendocrinology should be appreciated, and with this, physical activity and crucial neuroendocrines that pre-transplant populate amongst the pre-transplant population should also be encouraged in context of further investigations. Physical activity investigations may be important factor in preparation for patients awaiting transplantation. It was Fry and team in 1992 who investigated the in vitro responses of leukocytes following acute anaerobic physical activity over 24-hour recovery duration, and the investigation informed and suggested that the reduction in proliferative lymphocyte response is and was a short transient one. It was Shepherd and team in 1994 who observed interleukins as part of their work. Interleukin-1 secretion may enhance the responsiveness of peripheral blood mononuclear cells, but prostaglandin secretion decreases interleukin-2 production. Graded sort of physical activity reduces the adverse immune reactions associated with challenging physical activity. It was Wainwright's and team in 1999 who informed through a comprehensive review and considers the literature on across 6 aspects of recovery and rehabilitation. So, for example, the psychiatric sequelae, the functional recovery, stress and coping, the psychological impact of failed renal transplants and quality of life, and or QOL inside are aligned to adherence with particular forms of medicine. The team conclude or inform there is a pressing need to move away from descriptive research toward carefully designed multidisciplinary investigations with transplanted recipients. Physical activity elicits an increase in the numbers of circulating lymphocytes and lymphocyte subsets, including natural killer cells. T cell proliferation and T cell production of interleukin 2 and interferon gamma is reported immediately post acute intensive physical activity. The cumulative effects of repeated bouts of intense physical activity cause a consequent elevation of stress hormones, particularly cortisol and anti-inflammatory cytokines. So, for example, interleukin 6 and interleukin 10. This causes a temporary inhibition of type 1 T cell cytokine production with a relative dampening of type 1 cell-mediated response. It was Starkey and team in 2001 who examined the effect of physical activity and androgenic blockade on lymphocyte cytokine production. In investigations where there were six males or 6 men where they ingested either a placebo or control or an alpha prozazine prozazine hydrochloride and a beta androgen receptor antagonist, a beta androgen receptor antagonist blockade 2 hours before performing 19 minutes of physical activity. The physical activity in the literature was bicycle based physical activity. Blood was then drawn and collected before and after physical activity. So the surface stained for CD3. So blood surface in around under laboratory conditions was stained for CD3 positive and natural killer CD3 negative lymphocyte surface antigens, intracellular cytokines, interleukin 2 and interferon gamma, and then analyzed using flow cytometry. The investigation informed that blockade had no effect on resting concentration of stimulated cytokine positive T and NK lymphocytes on the or the amount of cytokines that were being produced. It was Starkey and team in 2001 informed that physical activity does not appear to be responsible for the physical physical activity induced suppression of cytokine production. It was in 2001 and Stensberg and team where in response to physical activity, both CD4 and CD8 T cells were mobilized to the blood, but the levels of these particular cell types declined below pre-physical activity values in post physical activity durations. Ipfelt in 2002 demonstrated that physical activity induces a decrease in the percentage of type 1 T cells. The investigation informed and demonstrated that physical activity induced change in type 1 cytokine producing T cells is related to a decline in memory cells. Imrich and team in 2004 informed that repeated stress-induced elevation of epinephrine and norepinephrine was not associated with an alteration in immune cell redistribution found in response to the single bout of physical activity. Mohammed in 2006 informed that using a multidisciplinary research effort would prompt professionals to come together to achieve a better understanding of in aspects of psychology, endocrinology, and immunology in order to achieve transplant tolerance. And it was also Mohammed in 2006 who attempted to answer questions in previous works in relating to the immune system, transplant rejection, and psychological stress. In and around this, as part of the discussion and conclusion, I'm now going to So in and around the discussion and conclusion aspects, I'm also going to link some of the discussion and sort of conclusion aspects in relating to particular questions. So, one of the questions here is how do studies, investigations in physical activity relate to transplantation? And so is physical activity a contentious concept with transplantation and stress? And so before answering those particular questions, the questions one of the particular main questions is how does how does psychological stress associate with transplantation and neuroendocrinology? So the neuroendocrine system generally regulates things that are most important in controls, for example, reproduction in all its aspects, from bonding to intimacy, to control of spermatogenesis and the ovarian cycle, parturition, lactation, and maternal behavior. It controls the way individuals respond to stress and infections. It regulates metabolism, influences consumption, and influences how energy intake is utilized, and etc. It influences mood. Also regulates body fluid and electrolyte hemostasis and arterial blood pressure. So depending on the state of an individual, for example, there can be good coping and stress adaptation mechanisms, and then in this regard, an individual can then, or at least from a physiological standpoint, there is a secretion of what is referred to as good hormones. Having a negative mental attitude in pre-transplant durations may have an effect on post-transplant outcome, leaving the allograft prone to chemokine receptor migration. Whilst glucocorticoids and monoclonal antibodies are important and suppress certain immune responses, i.e. diminish the amounts of interleukin-2 and lymphocyte cells to be activated, the neuroendocrine system is of central importance to many problems that are in regards to major health concern. However, as yet, there is no sort of formal evidence in context where there is a link between stress and chemokine receptor migration in context of acute allograft rejection episodes. In this original works, there's an overview of cell surface antigens for immunosuppression therapy, or ISTs, and the schematic is sort of an overview of the of antigen presenting cells and T cells, and where there's a link between cell surface targets of biological agents, and these are in context of different immunosuppression therapies. And so more research linking immunosuppression therapies, monoclonal antibodies, and cell surface targets for different drugs, and whether specific hormones affect chemokine receptor migration are in some contexts still required, and also to identify is there an association in the first instance, or in the first place. Psychological stress is associated with aspects of neuroendocrinology, but the transplantation aspect is subject to debate and criticism. So further research in this area is required, and a link between the psychoneuroimmunology psychoneuroimmunology is also called for, simply because the whole body needs to be observed and not just single organ systems. So in regards to another particular question that this works seek to provide some basis around was immunology versus neuroendocrinology. What does this mean? So during the past a decade, if not longer, the association between stress adaptation and the immune system has come under closer scrutiny, and there is now considerable evidence that certain types of experimental and naturally occurring stress are associated with alterations of cellular immune function. Furthermore, these observed changes are generally immunosuppressive. However, the mediating mechanisms underlying this relationship or association remain poorly understood. Critical factors in stress adaptation, immunity, and relationship are the duration and proximity of the stressor, the adaptive capacity of the individual, and the differential effects of certain stressors on immunological components. And so increases of circulating glucocorticoids after infection, for example, and an apparent activation of cerebral catecholaminergic cells indicate that challenges to the immune system are interpreted physiologically as stressors. And moreover, they suggest or inform that the brain may be able to monitor the progress of immune responses. Experimental and clinical investigations inform or demonstrate that both laboratory and natural stressors alter the activities of lymphocytes and macrophages in a complex way. And these, for example, can include number one, the type of immune response; number two, the physical and psychological characteristics of the stressor; and number three, the timing of stress relative to the induction and expression of the immune event. In this case, the time of in this context, the timing of transplantation versus the beginning of acute allograft rejection episode. In line with this as well, the influence of stress on immunity are mediated not only by glucocorticoids but also by catecholamines and pituitary hormones such as growth hormone. The immune system receives signals from the brain and the neuroendocrine system through the autonomic nervous system and the hormones and sends information to the brain through cytokines and chemokines. These connections appear to be part of a long loop regulatory feedback system that plays an important role in the coordination of behavioral and physiological responses to infection and inflammation. And there is much still to know about how stress and mental well-being affects the immune system and on an onset and progression of disease. So the sort of follow-up question in relating to this is how does this relate to transplantation? So one of the focal points of this sort of branch of this branch of sort of clinical science or clinical medicine is its ability to link stress and disease. Generally, there are now ways to pinpoint, and retrospectively through literature, etc., and through primary investigations, there's ways to pinpoint. There have been documented or evidence ways to pinpoint changes, for example, in hormonal flow, as well as various messengers and signal pathways within the body to and from the immune system. And so the stress hormones can change immune cell behavior and the activity of the entire immune system. More evidence has come forward. More evidence has been documented to project that there is a link between the immune system and psychology in context of the psychoneuroimmunology concept, and the P&I concept generally embraces that particular integration. And so it is anticipated that instigating some of the investigations that have been highlighted through the sort of research methodology aspects of this original works, that some answers become more transparent, and the link between psychological stress and transplant rejection, or at least acute allograft rejection, is still uncertain. Neuroimmunobiology holds the premise of treating immune and inflammatory disorders and the natural regulatory molecules such as hormones, cytokines, neurotransmitters, and neuropeptides. There is potential for this specialty to be linked with in and around acute allograft rejection post-transplantation. And so another question around this is, as sort of being documented, is can T-cell tolerance post-transplantation be achieved by a better understanding surrounding the immune system, neuroendocrinology, and psychology, and in context of psychology, mental attitude more specifically. So there is now evidence that in the same way different perceptions of stress have different psychological consequences, different ways of, for example, dealing with stress or coping with stress result in different consequences surrounding the immune system. So the nature of outcome of which depend on the type of immune response is one. Transplantation has also come a long way, but however, unfortunately, demand on renal donors on renal donors specifically is still generally a tremendous problem. And this is generally generally in context of a concept to link body and mind, and it is difficult to postulate outcomes because at the moment there are there are no sort of investigations which sort of link in context in their entirety. So investigations combining the immune system, neuroendocrinology, and psychology generally have been, at least in these original works, have been postulated as particular prospective investigations. And so another part of this is could a combined approach be important to attain T cell tolerance? So as this is generally a novel concept, at least in some regard, one has to have a sort of broad mind when looking through sort of the not least the literature, but also through different systems. So one may not be able to grasp the notion of the relevance of neuroendocrinology, sort of mental attitude, or even physical activity in context of physiology and transplantation. But the main point being made here is that all entities do have a connection, and that is in the investigation of leukocytes, lymphocytes, and their migration. There is a need to endeavor and see whether there is a connection between healthy pre-transplant recipients during and post physical activity and versus sort of mental attitude in and around a link and to see or identify sort of more formally if there is a link between leukocytes and lymphocyte migration. So if this could be investigated with more aligned to more neuroendocrine investigations, so for example cortisol, look at investigating cortisol, beta endorphin, and T cells, this would encompass various systems, and that is what is required in prospective research. This could be a potential way way forward to achieve T cell tolerance, and indeed indeed sort of stress adaptation and sort of coping mechanisms, along with the psychoneuroimmunology concepts or topics to contemplate for prospective topics surrounding transplantation as well. So in context of the sort of the sixth sense aspect surrounding the immune system, and so does the immune system serve as a sensory organ in context of the sixth sense? So the works by Wayent and team in 1990s in 1990, where the concept where they looked at the concept where the immune system may serve as an organ is an intriguing one. And the team in Wayne's team in 1990 informed that the recognition of such stimuli is converted into information in the form of lymphokines, monokines, and hormones, and a message received by neuroendocrine tissues. And on the other hand, nervous system recognition of other stimuli can be converted into chemical signals that can be relayed to immune cells, resulting in physiological changes. Prospective investigation into the physiological role of cytokines and neuroendocrine hormones have in these systems will be of considerable intrigue for both immunologists and endocrinologists. Yeah, so for both immunologists and endocrinologists, and it was Wayent and Blalock who surmised that sort of aspect in 1995. And so um, in context of the question, for example, why is it a challenge to attain T cell tolerance post transplantation? So it was Mohammed in 2006 informed this quite simply with the following. So in context of advances, the immune system is an effective immune system that it allows to be prepared and equipped for an attack or an invasion of the body. In context of physiology, and because it is an effective sort of system with cellular and humoral responses, it is a challenge to manipulate it to achieve tolerance. It would seem sensible that complications caused directly by, for example, immunosuppression therapy could be minimized if more neuroendocrine, immunological, and psychological investigations or studies are postulated. And so this could lead to a better understanding of pathways to prevent or preclude acute allograft rejection. And another question in line with this as well, should there be more efforts in the understanding of all systems, and typically in sort of in sort of in sort of a part of the original works documented. There is as sort of annotated, there is a schematic which outlines the general relationship association between the central nervous system, neuroendocrine, and immune systems with in context of health and disease. In essence, the answer to the above question is with regard to that schematic. So yes, there should be more efforts which link or incorporate an understanding of all systems. Laboratory studies or investigations are required, which encompass crucial immune components with neuroendocrines and stress cellular adhesion molecules that are required. Until methods are initiated, the tolerance the tolerance promotion sort of association or link, i.e., for example, what hormones and what type of stress are still unknown. In context of physical activity and physical activity being a conscientious concept with transplantation and stress, the answer around this is both yes and no. So yes, because there are no in-depth investigations linking the psychological and neuroendocrine concept with transplantation, and physical activity and positive hormones should also be considered in healthy pre-transplant individuals or candidates. So a no as part of the answer around this, because as with all research and literature reviews, nothing is definite. And so with regard to literature, the literature reviewed in context of this original works, methodologies proposed and questions put forward, and previous and some aspects of looking at the types of investigations, there are various ways that all these entities could be. There are various ways that that all these sort of aspects could be looked looked at in more detail. So now, in context of the conclusion around this works. So the hypothesis in this works was to identify whether one of the three systems, for example, the immune system, neuroendocrinology, or psychology, dominates in recipients awaiting an allograft, i.e. In and around the pre-transplant phase. Unfortunately, without studies or investigations to support the hypotheses, it is difficult to postulate whether there is a more dominant system. One can assume that both the neuroendocrine system and psychology can influence the immune system. Stress may be a factor which could pressure acute rejection episode or acute allograft rejection post-transplantation. Altogether, the objectives surrounding this work have been fulfilled, but until there is a collaborative approach and methods which incorporate investigations from the different fields outlined, acute allograft rejection is still a concern post-transplantation, and indeed stress adaptation and sort of mechanisms along with the psychoneuroimmunology aspects are topics to contemplate for prospective research surrounding transplantation. The physical activity entity the physical activity aspect also requires further investigation in sort of healthy transplant candidates to observe neuroendocrine and immunological chemotactic patterns. So in conclusion, at this time, the literature surrounding this work was neither for nor against the hypotheses, and probably debatable without full laboratory investigations as suggested. It is a challenge to state any particular direct correlations in context of specificity. And finally, a bit to encourage to read and review the works of Dr. Hans Selye and Dr. George Engel. So I want to say thank you for taking the time to listen in on this audio podcast surrounding this works surrounding transplantation, immunology versus neuroendocrinology and psychology. And this was a sort of an overview or an update in some regards based on works that have been sort of developed retrospectively from 2007. So once again, thank you for taking the time to listening on this. This has been the Shientist as part of this audio podcast in this particular series, the ZtormCrux Continuum, and this has been another Storm Robotics production. Thank you.